without further delay, I'll start my talk. As Islam already mentioned, today I will be talking about graph deep learning on healthcare applications. Before we get into that, I'll just briefly tell my background. So I come from India. I did my master's from Indian Institute of Technology, Kharagpur. And through a DARD master sandwich program, I was given an opportunity to do my master thesis in Technical University, Munich. And my major during that time was medical imaging and informatics. So from my master's, I am involved in medical image analysis and informatics with that. So I did my master thesis with Professor Nasir Nawab working on cross-modal manifold alignment. So basically also, since masters, I'm involved in manifolds and graphs and so on. So I really love this topic and I love to talk about it also. So I started my PhD in 2016, also at the chair of computer-aided medical procedure and augmented reality. The short form is CAMP, that is from Professor Nawab. And in the first year, I was working on general deep learning for medical applications, focusing on bone fracture prediction on X-ray images. I started working on graph convolutional networks, or per se graph deep learning, from 2017. And that was also my major topic for my PhD. During my PhD, I looked into multiple graph scenario, like how to deal with multiple graphs together, how to deal with graph heterogeneity, how to learn graph, how to do personalized disease prediction using graph deep learning, and also data imputation. As you might have seen, that's a very famous topic from Netflix and also from Twitter, like how to do data imputation or kind of get values for missing data. During my PhD, I also visited Imperial College London, and I was there for 3 months, wonderful place. And basically I worked on graph learning together with Professor Michael Bronstein. Shortly after finishing my PhD in 2021, I started my postdoc, which was a short postdoc thanks to COVID. I didn't want to move anywhere. And it was a wonderful opportunity for me to serve as a postdoc for the same chair where I did my PhD. And I focused on multimodal data analysis and graph deep learning during that time. During my postdoc, I specifically worked on cross-modal data retrieval using graph convolutional network, graph classification, image registration using GNN transformers, a little bit of EEG signal analysis for epilepsy prediction, and then interpretability of graphs. Now, during my postdoc at MGH and Harvard, I'm focusing on brain connectome analysis. I started my postdoc here last year in the month of September, and it is an ongoing project right now. In terms of publications, or let's say the specific works, we looked into together with the whole group of graph deep learning at CAMP and also all the collaborators all around. We looked into handling missing data, then how to basically increase robustness with multiple graphs and self-attention together, how to basically do, as I mentioned in the previous slide, personalized medicine or patient-specific attention to kind of do better diagnosis and treatment planning. Then Inception GCN, which is one of the very good paper, at least from my pool of papers. Then we also looked into how to get CNN and GCNs together. So what if your data set is like an image data? So how to give image as input to your data set. Then we looked into how to incorporate text prior knowledge to get better predictions using graph deep learning. Then, of course, population graph learning, knowledge distillation. This work was done together with one of my PhD student, not my, but Nasi's PhD student. I was helping her finish her PhD. So looking into knowledge distillation, metrics completion, and then basically extending our previous works to different journals. I was also involved in a COVID project at CAMP, where we kind of looked into how to predict whether the patient is going to be admitted in ICU or predict mortality, given the situation, given the features based on the chest X-ray and all other clinical features for COVID, how basically you can predict whether the patient will survive or go to ICU and so on. Also, as mentioned before, cross-modal image retrieval or data retrieval. So basically, given two manifolds, how can you use one manifold to get the data out of the second manifold? We worked on protein interactions. This was continuing our collaboration with ICL, that is Professor Michael Bronstein and Professor Dr. Luca Cosmo. Then we worked in brain signal analysis, that is dynamic gated graph rectifying unit. We developed it and then published it in SPIE 2022. We also worked on reweighting attention graph, reweighting adversarial graph convolutional network. So basically, when you have missing labels and missing graphs, how do you deal with that? We recently published a paper in ICLR 2023 on product manifold. So product manifolds, so basically including hyperbolic metric, spherical metric, and so on into the picture to learn the graph, and also looking into graph transformers to predict like future features of a particular subject, for example. Now I am more into GNN explainability, that is also interpreting GCNs, to get better interpretation of your input data, like which input features are more important for your prediction, for example, and of course, brain connectome analysis. In brain connectome analysis, we are focusing on classifying the brain graphs currently based on their diseases, based on their gender, based on their age, and so on. So it's a basic problem so far, but designing the model is super exciting for me. So today I will talk about these four methods. Of course, I won't be able to cover all of them, but please feel free to reach out to me if any of this method seems interesting to you or is related to your work and you would like to discuss about it. I'm up for collaborations and discussions. Okay, so let us start with our first work, that is DGM, that is differentiable graph module. I'll talk about it. But before that, let us establish a bit of the background, right? So let us say you have a population level graph where each node is a subject. Each subject comes with a feature vector that is coming from medical features. Let us say if it is a brain disease prediction problem. So the features come from MRI segmentation numbers, or clinical data such as demographics or genetics. Also, sometimes there are tests like MOCA or some kind of clinical tests that are being taken from the patients, scores of those tests, and so on. So consider that these features are multimodal features that are collected for each subject, and in this graph setup, each node is a patient which is associated with such a feature vector. We connect these subjects also with certain criteria, right? So that is another topic, but let us say that we connect them by having a distance matrix between these features itself. And EIJ basically denotes whether there is a connection between two subjects or not. And WIJ denotes like what is the weight of that connection. So how strongly two subjects are connected, right? And our model is as simple as this. Given the input features and the graph, our graph convolutional network has to predict the class. Here, let's say our problem is classification. So basically, this problem turns into node classification problem. We want to classify each node or each subject into a particular class, right? Now, the first question is, what if we don't have this graph at the first place, right? So not all the data sets come with graph, right? And in the state of the art, at least at the time when we were working on this problem, the state of the art networks were creating this graph in the pre-processing step based on demographic information or based on, as I earlier mentioned, that distance between the feature vectors of each subject. But we did not know whether this graph is optimal or not, right? And often in a couple of our preliminary analysis, we also realized that that graph is not optimal because, for example, if you are creating this graph based on gender, you basically get two different clusters. But sometimes gender has nothing to do with the disease that you're predicting. So in that case, the message passing that happens in the graph kind of degrades the performance even further. So we thought that why not learn the graph based on the task and based on the input data that you have, right? So this is the first work that I wanted to talk about, that is a differentiable graph module. The name of the paper is Latent Graph Learning for Disease Prediction. So given the subjects, input subjects with their input feature and no graph, how are you going to learn a graph from this, right? So what we do is we take all the input features of each subject and send them to a function f of theta. This function is really a generic function. It could be an MLP. If you have like vector features, it could be a CNN if you have image features, or it could be literally anything based on what kind of input data you have. So we get an embedding space based on these input features. And in this embedding space, basically, we kind of try to generate the graph. So what we do, we use like L2 distance between the feature vector of each subject and create a fully connected graph out of it, and then send it through a sigmoid function. The threshold of this sigmoid becomes our learnable parameter in our network. So this becomes a soft thresholding, right? So basically we get a graph which is sparse as an output, and this graph is then sent to the GCN. This GCN could be any generic GCN module or model of your choice. And we take this input graph and then the features that were again given to us during the input time, and then do the general graph node classification task with this GCN. The good point about this model is that it is, you can back propagate through this entire process, right? Given that sigmoid is differentiable, your MLP or whatever f of theta function that you're using, given that you're using differentiable function, is differentiable. So basically it is possible to then train this upper part of the network also, which is why we were able to kind of, for every epoch or for every training, we were able to optimize our graph over on fly. So it is also scalable. So this is inductive. So what we do is we take the learned weights of this function, and if a new subjects come into the picture, we just embed that new subject into this embedding space and calculate the distance of that particular new subject with other subjects, and then proceed further with our classification. We applied this method to three different data sets. So for tadpole data set, which was like Alzheimer's disease prediction data set, and we got like 7.4% increase in terms of accuracy of classification compared to state-of-the-art networks. And in inductive setup also, we got like 9.18% increase in accuracy. Same happened for UK Biobank data set for age prediction task. We were able to increase our performance compared to state-of-the-art with a very good margin. We applied this method also to a computer vision task, that is point cloud segmentation. This is a huge data set, and on top of that, it had like each point cloud had to be sent in the network separately. Here we were sending the entire population at the same time, right? So we also wanted to see how our network works when each of the shape has different graphs in itself, right? So there also we were able to increase our performance by really small margin, 0.4. But when you compare this with the size of the data set, it is a significant increase in performance. So we were able to push this work to MICCAI 2020, and then we extended it to our journal, which was published in TPAMI in 2022. In our journal version, we have a lot of ablation experiments, which are really interesting. So basically that will give you insights of when the model breaks or when it works, given the input data set and also the task at hand. So I would recommend you to definitely read this paper and also tell me if you find anything, anything that is contradictory or as I can work on, or, take it forward. So this was the first work. The second work that I want to talk about is interpretable attention. So basically, now we have looked into how we learn the graph, right? So we wanted to look into the effect of node features itself too. So given the input setup as it was from a graph constructed from medical features, node features definitely coming from medical data. So basically we wanted to see which input feature is important for the task. So here, this is the name of the paper, that is Interpretable Attention-Based Graph Convolutional Network for Disease Prediction. The aim was to develop a simple module that can track back the important set of features for a particular task. For example, the task could be age, gender, disease prediction. Given these three tasks, which input features are most relevant? And this was also like interpreting the input setup, like in terms of clinical interpretability of the model, right? So what exactly we wanted to do, as I explained earlier, given the input set, let's say you have 10 D features and you just want the model to let you know which ones out of this are more important. So given a GCN model, as we saw earlier also, which takes graph as an input and node features as an input and gives you a label, right? The direction talks about the forward propagation right now. And we assume that this graph is either given or not given. We also have in our journal extension of the paper where the graph is given, but here I'll talk about when the graph is absent, because I just spoke about how to learn it, right? So you take, so basically this is the graph convolution. I'll skip this slide. You take the graph learning module, that is DGM, which we just spoke about in the previous slide, and push your input features to it and get this graph. And get the same feature and push this to GCN and do your label propagation, right? So this is the same model that we just discussed in the previous work. Now, what is new that we wanted to do on top of this? So we developed this interpretability attention module over here. So what we did is we just initialized a mask, which is of size of the input features, and sent this input mask through a sigmoid function. So basically here you will have like a weighted mask. And this mask would be then multiplied to the input features together, and then the weighted input features would go into GCN. Until now, this is still not super exciting, right? All we have done is basically just had a mask, and then we are pushing it through some function that is going to learn the weights, and then weighted input is going in. The important part comes in the back propagation. So here you can see we developed a customized loss, that is interpretable attention loss, which actually is just sent through the classification network and your interpretable attention module. And your general loss, that is cross entropy loss for classification, is sent through the entire network as it is, right? So here, these were the two things that were new compared to our previous work, and basically the loss. So in our loss, interpretable attention loss, we had two terms, that is feature mask entropy loss and feature mask size loss, that was doing the magic for us. So the feature mask entropy loss basically was pushing the value of this elements of our mask away from 0.5, which basically increased the confidence of the model, or increase the confidence of this interpretable attention module to tell us about which feature is more important, right? And this particular term in our loss function basically tried to reduce the weights of all the elements in our mask. So what happens is that if you combine it with the cross entropy loss term as well, so this particular loss term tries to increase the value of the masks all together. However, these two kind of try to reduce the value of the mask and also send the value of the mask away from 0.5. So which is why this mask started becoming more and more confident when we were training it. Do you have any question, Islam?
I was just commenting. I said this could be considered as a regularizer of the learned attention mask. Yeah, these two extra terms that you constrained your learning.
Yes, exactly.
That's so cool.
So basically, we applied this method to similar data sets because we wanted to compare it with our previous work, DGM. So we applied it to Tadpole and UK Biobank dataset for Alzheimer's disease prediction, sex classification, and categorical age classification. In all the 3 datasets, the model was outperforming the previous methods with good margin. We also kind of did an ablation of if the two lost terms that we have added, they make sense or not. So basically what we did, the alpha term, I'll just show you again. So alpha is here, right? So we kind of removed completely this term by putting value of alpha equal to 0 and gradually increased it and put value of alpha as 1, removing literally cross entropy term by itself to check how much effect this particular lost terms have on our entire training. So you can see that, of course, when we remove complete alpha, the accuracy, basically this is like if we keep alpha is equal to 0, that is only training on the 2 lost terms that are not the cross entropy terms, but the 2 lost terms, that is interpretable attention term, the accuracy drops to 0.5. But as you gradually increase alpha, at 0.6, we get like the optimal value of our accuracy term. I think here the alpha has to be other way around. So 0 alpha means that the 2 terms, sorry about that mistake. However, what I wanted to say was when you put the value of alpha, that is the to keep only 2 terms, you get like accuracy really low, which means that cross entropy term is really important. But at some point, the combination of 2 gives us the optimal result. We also performed an experiment where we kind of kept all the features that are selected by the model and removed all other features that are not important. A, B, C, D are actually different setups of input features, and D is the one that is important to look at. So you can see that the features that our model selected, when we fed those features to our only those features to our model, it was outperforming really well compared to other setups. So this is the second work. And we also checked the features that were selected by our model. So basically CD, BRS, and MMSC, both of them are clinically considered as really relevant features for which correlate very well with the labels. So in conclusion, we basically generated a generic interpretable attention module. A graph learning module was already there and a unique loss function. So our model learns the attention for input multimodal features and uses the generated interpretations to train the model. So actually IIM can also be incorporated in any other deep learning model together with the task that you want to perform. And features selected by our model had high correlation with the ground truth for Tadpole dataset, that is the cognitive test features. Next work that I want to talk about is actually done by Masa Gorbani. So she was the PhD student that I used to work with during my time in camp. And the work is RAGCN, that is Graph Convolutional Network for Disease Prediction Problem with Imbalanced Data. So here what we are trying to focus on. So let's say you have a completely imbalanced data and the data distribution looks like this, right? So what we did is we trained an unweighted MLP and checked how the classifier looks like. So this unweighted MLP gave us 88% accuracy. But when we looked into the macro F1 and binary F1 score, we saw that it is just 0.49, right? Even ROC, AUC was 0.5. But if you just look at accuracy, it looks like the classifier is performing really well. However, it is literally failing, right? So basically, one conventional way to deal with this is to have weighted cross entropy, where you have like weights that are inversely proportional to the frequency of samples in the class, right? And these weights are pre-computed and they are not sample specific either. So basically, each sample in one class will be given one particular weight. Now let us look at an example where the data distribution looks like this. The circles are class 1 and class 0, and the squares over here are class 1. Sorry for the really not very clear description of it, but we wanted to show like the confidence of the model while predicting each point in this particular distribution. So basically here, if we take a point on this side, the classifier classifies it as class 0 with a confidence of 94%. And if you take a point B, which is close to the classifier kind of has a confidence of 52% for classifying it in class 0, right? So in our data distribution, the position of the point is also important during classification. But in case of our conventional cross entropy loss, both this point are given same weightage, right? They are pre-computed based on the frequency of the class, and basically it does not matter how much this point affects the classifier and how much this point affects the classifier. Both of them are important. So this particular point on the left is important to learn like really good representation for that class, right? How a feature vector, a learned feature vector for this class should look like, and how and this particular point is important while defining the line of classifier, right? Because it is the most confusing point. So both of them are important in a different way. So we wanted to focus on that point in this work. So we kind of developed a dynamic weighted cross entropy loss, where we tried to learn the weight for each point in our data set. And we trained this whole dynamic cross entropy loss using adversarial training. So we were minimizing the whole loss, whereas we were maximizing the weights over here. The model looks something like this. So you have an input graph with input node feature vectors and all the nodes also, and the task is to do node classification. The upper part here is our conventional GCN, right? So basically couple of GC layers, you just send this input graph and the node features, and you get like class probability at the final stage. The novelty lies in this lower weighting networks. So in this lower weighting networks, what we did is we sent all the training samples as an input, and each branch was dedicated to one particular class. So we would segregate the input nodes that belong to each class and create the graph within themselves. So what do I mean by create the graph? We would not create new graph, but rather just the points that are already connected in here and belong to the same class would be connected in here, and we will create a graph like that. And here also we'll have a couple of GC layers, and the main objective of those GC layers is to learn the weights for each node. How this will happen? So basically you take input node features also, right, together with the input nodes for one particular class, and you'll learn the representation for that node using graph convolution. And at the end, for each node, the one value as a representation that it is learned, we use that as the weight value basically for that sample for that class. And then in our objective function, we have the cross entropy loss, where the weight is basically learned, so dynamic weight learning loss function, and also an entropy term to avoid like sharp distribution of our weights over here. So this part is basically the regularizer. So this whole model we train end to end in order to learn sample specific weight for our data sets. And definitely graphs help us here, and also in the upper part where we are trying to learn better representations for each node based on the connections. We applied this particular model on 3 data sets, that is diabetes, PPMI, and Haberman. You can see that the data sets are not super large, and also that input node feature distribution is also super different. Like here we have 300, whereas the other 2 have only 7 features, so not a lot to learn from also. And number of features for each graph that was used was also 4, 1, and 1. And basically we also kind of showed here the adjacency density, that is how dense a particular graph is, and the imbalance ratio. So you can see that at least diabetes data set is quite okay, but however, the other data sets are imbalanced. So the results on diabetes and PPMI datasets are here. So we compared them with MLP non-simple cross-entropy loss, MLP weighted cross-entropy loss, generic GCN with unweighted and weighted cross-entropy loss, and a state-of-the-art network that is DRGCN, and then ours that is RAGCN. And we should look at mostly macro F1 and AUC ROC AUC numbers. So here you can see that our method was performing much better than other compared techniques. And similarly for PPMI dataset, so DRGCN was performing better in terms of ROC AUC. However, our method ended up performing better for macro F1 score. For Haberman dataset, it was a bit of a different result. So our method was performing really good in terms of accuracy and macro F1, whereas it was performing comparable. As you can see, the difference is 61, 65 in terms of AUC, ROC curve. So in conclusion, we then saw the classifier, how exactly it looks like, right, after training. So left-hand side, you can see the figure for GCN, where we have trained the model using weighted cross-entropy, and right-hand side is our model, that is RAGCN, where we have trained it with our customized cross-entropy loss. So one thing to notice here is definitely this line itself. So as you can see that now the classifier is no more misclassifying the samples, but rather it is kind of adjusting itself to a much better location in the distribution. So this was the work RAGCN that was included in Masa's PhD thesis, and yeah. So next work that I want to talk about is knowledge distillation. So basically this work also comes from Masa, and we published this in MICCAI 2021, and the name of the paper is Semi-supervised graph knowledge distillation for graph independent inference. So as usual, the problem is that you have node feature vectors, adjacency matrix, and a graph. And you have a function f, which takes the graph and the input feature as your input, and then gives you like a lower dimensional embedding, and you have to classify the lower dimensional embedding into the class, right? So what exactly is special about this method? So first of all, during training, you have a training set, but you have very limited labels. Like this is first problem that we want to address in this work, like how to deal with a data set that has very small label. And I think this is a very important problem in medical field, right? So you don't get a lot of labeled data sets in at least in medical domain. And for test data set, this is actually test data set, you have no labels, and of course you have no graph. So these are the two problems that we want to talk about in this particular work. So first of all, limited labels, but you have graph in the training part. And during testing part, definitely you don't have labels and no graphs also. So it is an inductive and semi-supervised setting. So what happens if the graph is not present for the test sample, right? So this is just a very simple pictorial example of it. Let us say that these two nodes are your test nodes, right? And you are sending this graph, which is only limited to training samples, and basically send it to any GCN network. And it kind of learns the representation based on the neighborhood of each node, right? So here the aggregation color of each node also represents like how the feature or message passing will happen. So what we should look at is that message passing did not happen to the test sample at all, because there was no connection. So if you try to then classify them, it's basically not using any graph data. It is not using any information from training sample as such. It is indirectly using information from training samples, because the weights that are getting learned during the training using the training graph and features, it will affect the test nodes, but not due to message passing. So this is the input setup. You have like training samples. The gray points are the points that are training samples but do not have any labels. Red samples, the colors represent the class of the other training samples where the labels are present. And at the test set, only you have only the feature vectors, but you do not have any graph within them. So let us just concatenate them together. So what we want to do is we want to have a network that takes such a setup as an input and does your task. So here again, our task is to do node classification, right? So what we went for is a teacher-student network. So the setup was like, first have a teacher network that deals with the input setup that you have in your training set, and then use a student network that deals with the setup that the test samples have, right? So a teacher block, teacher's network looks like this. It is really, again, very simple. So you take an input graph and you take features of labeled and unlabeled node together. You send them to a teacher network that I will describe in the next slide. And you try to generate pseudo labels for unlabeled node that are there. So in different words, you try to basically learn the labels of the unlabeled node during training. So indirectly teaching the teacher first in a way. So basically what you do is we call them pseudo labels that are coming out of this teacher network. And then you have a bigger pool or bigger labeled samples, right? And then you go for the student network. So the student blocks looks like this at the bottom. Let us focus at the bottom. So given that the teacher network has given us the pseudo labels for unlabeled nodes, we take those pseudo labels and basically use those pseudo labels as our ground truth in our student network, and then take the test samples and push them through the student network, and then train the student network and teacher together. Now, we also said that we don't have graphs during the test time, right? So here our student network basically won't be a graph convolutional network. It will be a simple MLP that will take your input features and try to classify the test samples with only their input features into respective classes, right? So now the elaboration of the teacher network. So you have the set of labeled node and unlabeled node, right? So we take like a fully connected network and just predict the labels of all the samples over here. So it's like a classification task that this fully connected network is going to do, right? We train this fully connected network using cross entropy laws and train it back and forth, of course. Now, after some iterations, let's say after T1 iterations, we basically have a set. Let's still segregate them, where this is our labeled nodes, where the ground truths are literally 100% and the other classes have 0% probability. Whereas these unlabeled nodes, they are still a bit fuzzy, right? We don't have exact 100% confidence from the network for their label. So the distribution of the label for the unlabeled node in the training set still looks like this. So this is where graphs comes into picture. So what we do, we take the training graph, right? And we build the graph between all these nodes, and then take these labels and this distribution together as their initialize them as the node feature, and perform a label propagation algorithm on top of that. So what is the label propagation algorithm? So let us call these distributions as your FC outputs, right? As your output of your last layer of your network. So the nodes which actually had labels already, we don't want to modify them. We don't want to disturb them. So we just kind of re-establish the same label all over again, and not take the FC outputs for those particular nodes where the labels were already present. However, for the nodes which did not have labels, we basically apply this formula where we multiply the labels of multiply the adjacency matrix and the FC output together in order to pass the label information to each unlabeled node over here. And in this process also, the labels of the original labeled nodes get modified, right? So we kind of replace them with true label again. And we perform this mechanism after T1 iterations, T2 iterations, and then train the teacher network like that in order to make these labels more confident. So as an output of teacher network, we kind of get now labeled nodes, right? So instead of having completely this setup, we have a little bit better setup where the unlabeled training nodes have better labels now. All right, so then what we do, we use this new labels for our student network, as I mentioned earlier. So this is how the entire network looks like. You have your input labeled and unlabeled node, you train the network, you get the labels, and then you kind of do the label propagation algorithm over here, get the pseudo labels, and then use the pseudo labels in your student network. And then the test samples come into picture. In the student network, we do not use graph. So the job of graph is limited to this part of the network, that's it. And then for the student network, we just have like, again, a FC network. We take our test samples at input as input, and then all the learned labels and the original labels as input, and then train the student network as well. So basically this whole network is trained in steps. So it's a full end-to-end pipeline. So it's not like we're training teachers network first and student network later, but like in iterations. First train T1 iterations teacher, and then student, and the teacher, and so on. So this was the teacher-student setup that we worked on. And we again applied this network to a byte data set this time. And as you can see that our network was performing really well compared to all other state-of-the-art networks that we compared with. So we also applied it to a much larger tadpole data set. So the tadpole data set that I was talking about in previous couple of slides, that was a subset of actually tadpole data set, which had like 800 samples. But this one here, we all included like their multiple visits and also like other samples which did not have couple of features. And then the entire tadpole data set size was like 229 cases. And we saw that our method performs better compared to all other comparative methods also in case of tadpole dataset. So basically, that's it. So in conclusion, so today we looked at 4 different networks, that is DGM, that is latent graph learning for disease prediction, how to learn a graph. Second one was how to build an interpretable attention for your node features. Third one was how to tackle problem of imbalanced data. And last was how to train your model when your labels are limited, as well as when your graph is not available in the test time. Thank you so much for listening. I think I'll stop and take questions for last 15 minutes.
Yep. Thank you very much, Anees, for this fantastic talk and amazing, honestly, line of works, like very, very diverse. And I think we have so many meeting points. So I will start with a few questions. So here, generally these models, they work on so each node in your graph so far represents a sample, right? Yes. So basically the graph is a population of subjects or a population of samples. So have you tried to generalize, or have you tested all of these models directly on graphs, which means instead of having a population of nodes, basically you have one sample represented by a graph. For example, the brain connectome is a very good example. And instead of, building a population, doing population-based learning, you do a graph-based learning. So you have batches of graphs that you feed into your graph neural network. So do you think that these models can be easily adapted or to graph-based learning, which means you learn on the whole graph itself, or you have batches of graphs if you want to classify a whole brain graph, et cetera, yeah?
Mm-hmm. So really good question. Thank you so much, Islam. So one of the work that I actually did not discuss here was graph in graph. So there the setup was exactly as you mentioned. So each node was a graph. And then we kind of built a population level graph on top of these node level graphs too. So in that case, it was a two-step pipeline. First step is basically dealing with the node level graph itself. You send them to like a graph processing pipeline, which kind of takes the entire graph, does the node pooling or other mechanisms, and bring them to a representation, one representation, and then build a population level graph on top of that. So first two works are definitely translated to that. Like
cool.
we are working on it.
So you can bridge these both ends basically. I think all of these models could be easily adapted to be trained on, graph-based data. So without, without extracting just the features from an image, if the data, if the sample is a graph, I think they could be easily adapted. Cool, great. So there's a lot of potential there, definitely. So I do have another question about DGM, which I find really simple but fascinating. That's a marvelous word. I always tell my students, I always tell learners, you guys keep your ideas simple but formalize it very well, and you'll be surprised at the results you may get and generate. So could you maybe tell us a little bit? So I'm going to ask a bit of a design thinking question. So how did you get the idea? Like maybe if you go back and think about, okay, so everybody's using GCN, how did you come up with the idea of trying to learn the graph itself? Why, using, a simple, like this simple mapping function? Have you tried other things? Like, tell us a little bit about behind the scenes, because this is a very exciting work. So if you have any behind of the scenes, things to share, tips to share, we'll start with that, and then I'll follow up with a more scientific question.
Sure. So again, a very good question, actually. So basically, when I was, I read a state-of-the-art paper, as I was mentioning, that they kind of built the graph in the pre-processing step using demographic information, right? But when I switched the data set, I fell with the question like, okay, is this like the right choice for me, for my data set and my task? So of course I cannot answer it without doing the experiment. So I just created 4 different graphs on 4 different demographic information, that is age, gender, genetic, I think APOE was the value, and then one more was like a average, it was called like this cerebrospinal fluid number, like average value of that. And then I noticed that each graph structure was definitely different. And basically, when I was using each of them separately for the for my GCN model, every setup was giving me different result. So I was confused with, okay, which graph now is good? So if we kind of look at it in terms of publication point of view, we will just go with the one that performs best, right?
Yeah.
But if you try to do clinical interpretation of that, right, whether this makes sense. Like, for example, in that experiment, I was doing Alzheimer's prediction. And my graphs were age, gender, genetics, and the FDG value, right? So age was giving really good performance, probably because the age graph was correlated to the task that I was doing. But this is like a homework that we have to already do, like reading some clinical papers to find out whether the factors or features that we are taking for graph construction make sense or not. So we realized that probably it's a much more complex setup than that. So we are sure that the age is not the only factor, only risk factor, right? Sometimes it is genetics also, sometimes it is gender also. So how to mix these graphs together now? So the next step was like, how can we combine these graphs together? So we were like, okay, let's have a layer that learns the weight for all these 4 graphs, and then gives you a combination of the 4 graphs that becomes one graph later. So then we were like, okay, again, in this case, we are forcing the model to have fully connected graph every time. How to make it sparse, right? So everything is like, then we are setting it up, like how the threshold of the graph, the combination weights were definitely learned by the model. However, we realized that why not let the task decide it? Why not let the model backpropagation decide it, the threshold and the combination of all 4 of those? So we tried a setup where instead of having this skip connection, we basically had these features as an extra input here, but for the graph learning part, we only had those 4 features that we thought are important for the task. And then this model was kind of doing the combination of that together and learning the graph. So the question of learning it came from a lot of experiments and questions for ourselves, like whether what we are doing is right or not, like whether it makes clinically any sense or not, right? So yeah, it was more of a process of getting to
process.
The interest.
I love how you walked us through that. So thank you so much, because actually generally we read papers and we find like we are mesmerized at how the ideas that are published, but we don't understand the etiology, how these came into play. So you start with a preliminary experiment, you listen to your data, then you move from one question to the next question, and this is how you guys can design amazing things, particularly in AI. So asking the right questions, I think, is so important, and knowing your data is also so important. Great.
I think one more thing that is more important is the state of the art. So when we got to the question that we need to learn the graph, I think the same year there were like 2 papers that were working on exact same problem of learning the graph. So basically, if those papers were already there, probably we would have had some basic model to start with. So don't miss the state of the art papers at all. Like, read them all. They give you even better ideas to how to solve your problems. Much better.
Absolutely. Great. So I do have another question about the inductive setting, training, because you mentioned transductive and inductive experiments that you ran using this model. So can you explain how you trained and tested, especially for so the transductive part is kind of pretty obvious because GCN is transductive, but what about the inductive part? How did you make this work in an inductive setting where you can test it directly on new coming samples?
Mm-hmm.
So especially at the GCN stage, at the classifier, because for the student network is very obvious. You didn't use GCNs there. For the student, you had like a simple classifier in your knowledge distillation. But for this one, you have a full...
okay, sorry.
Oh, yeah. So it's about this one. Yeah, not the student one. The student one, I think, is kind of clear. Yeah.
So here, basically, a GCN will take a given graph as input, and then basically learn the representation as an output, and you classify the output, right? So we want to come to a stage where we have this graph for test samples also. Then the problem of GCN is not a problem at all. You will use the pre-trained model that was trained before, or basically also kind of just use the weights of the pre-trained model in this GCN part, right? So what we do is we take the test samples and come until the embedding state part. Here, f of theta is also already learned, right? All we want to do is just project the test samples to this embedding space. So once, let's say we have one test sample, what we do is we kind of try to create the row for that test samples in our affinity matrix, right? So once we just calculate the distance of all the samples through all the samples to that test node, and again push that test nodes row through the sigmoid function which was already trained, we have kind of a augmentation of the graph right here. So basically, then you just push this graph to your GCN and take the input features of the test samples and then just classify them. It is as straightforward as that.
Okay, cool. Yeah, so basically here, what you're using GCN for is like for another projection, for another embedding, right? Yes, so you're learning node-based representation. So you have these embeddings of the nodes, and then you just, feed the embeddings, the GCN embeddings through a simple classification layer, and that's inductive. Yeah, makes sense. Thank you. Great. So if we don't have any questions, anybody has any question, please feel free to raise your hand or, post them in the chat. But so yeah, I do have another question about the learning the weights, the attention mechanism you had in the class imbalance paper. So have you tried to just, predefine the weights? I was kind of, because there are so many, baseline naive approaches where instead of making them learnable, you can simply predefine. So this is exactly as predefining the adjacency matrix of your, graph, right, for your population. But in this case, where we're predefining the weights based maybe on Euclidean distance between the features, right? So have you tried to use a very simple naive approach and some before you went into learning?
So basically, in case of this particular function, you mean, so defining like a fixed
Yeah,
weight?
yeah, yeah.
So this, it's like comparing it with weighted cross entropy, right? Where we are defining the weight in the pre-processing step itself based on
Yeah,
like.
exactly. That's what I
So
meant. Yeah.
yeah, we did actually. So these
Oh, you do it.
unweighted and weighted are basically using weighted cross entropy. For our method also, we have tried, but we're not sure why they are not here in the paper. So basically the weighted and unweighted, we did try that.
Right.
The classifier was performing similar to weighted cross entropy loss situation actually. So basically it was no different than GCN weighted, for example.
Okay, cool. Great. So we do have actually a question in the chat from Ramona. She's asking about the multimodal data in the first study. So one challenge is the lack of this kind. One patient has an MRI scan but does not have, for example, genetic data. How do you think about this limitation? I think here it's the problem of incomplete data. So does your model handle incomplete data? Have you worked on the problem of incomplete data, learning from incomplete data, like where certain features are completely missing? Yeah, so I think that's the question.
Very good question. So in our method, we actually removed the patients where any kind of data set was missing. So we did not have that problem.
You discarded them.
Data, we just discarded them because that was not the focus of the problem. But there are two different works where we have worked on missing data problem. So one was matrix completion. So basically, again, having a graph and imputing the data based on neighboring nodes that are very much similar to your own node whose data is missing, and trying to interpolate that value. This is one way to go. Second way was using graph transformers, which is like also one of the very recent work, but there the data set was like a sequential data. So one patient had like 4 or 5 different time steps of data recorded, and one of them is missing, for example. So using graph transformers, we were trying to predict that one stage, given that we have all other data and labels at all the stages also. So problem of incomplete data is definitely a big problem. So it's not easily solvable. In order to kind of also impute good quality of missing data, let us say, we need like a lot of labels of the other available data,
Yep.
Yeah, so that's how we dealt with it.
Great. So one last question before we wrap up this session. So we have a question from Notice. She's asking about the weights, actually, the edge weights. So if you're not learning the edge weights between the nodes, right, and you're opting for predefining them, which kind of metrics or measure you have used and tested in your research so far? Like, I don't know, like, for example, she's mentioning correlation, maybe cosine similarity or Euclidean distance. So maybe you can tell us about the basic approaches a little bit, the ones that you have explored.
I'll just read the question one more
time. Yeah, go ahead.
Okay, so for the first network where we are learning the graph, we used L2 distance, simple L2 distance between the two feature vectors as such, or the learned feature vector as such. But in one of our other work, we are exploring like hyperbolic distances, spherical distances, and so on. So basically, this is actually a very good question. So Euclidean distance or any form of one particular distance may not be the great choice to kind of compute the distance between the graph. So which is why we kind of started trying like combination of the two. Combinations of different matrices together. So basically for some points, Euclidean distance may be really correct to define them, but for other distribution, it may not be. So my suggestion would be to kind of try the combinations of matrices together, like 0.5 times Euclidean distance, 0.3 times, hyperbolic distance, and so on and so forth.
Great. Thank you so much, Anees, again, for joining us today and for this wonderful talk. I think there's a whole lot to read, guys. This increases your reading list for the program.